This is the third tutorial describing Swift, our automated detection method for rare subpopulations in flow cytometry. In the first two tutorials, we guided you through installing Swift, performing a basic clustering of a sample, and then visualizing the type of output data provided by Swift. In this tutorial, we will explain how to compare samples, first of all making cluster templates that can be used for multiple further samples, making those from consensus samples, then assigning samples to those cluster templates, and finally putting together the output into usable tables of large-scale data. So one of the problems that occurs in flow cytometry data when comparing samples is that samples can often be similar but not identical in the positions of the clusters. For example, subject A and subject B look broadly similar, but if you look carefully at the positions of these clusters and overlay them, then you can see that the red and the blue aren't quite in the same places. And this can in some cases lead to difficulties with cluster matching. There is another more serious problem which occurs often in the type of data that we'd often deal with, which is antigen-stimulated cells. In a stimulated culture, we may have multiple populations present in both samples again, but in the unstimulated cells, one of those populations may be completely missing. And if a population is missing, or if there's maybe 3 cells in that population as here, it's very difficult to cluster that population directly. And so comparing clusters between samples that have been independently clustered is extremely difficult. So we have a strategy around that so that we can compare samples in a very rigorous way. And the first step of that then is to make a cluster template, and we first concatenate samples together that include all the populations that we're interested in. So we would use both positive and negative samples in the case of stimulated and unstimulated cells, and we'd make sure that the populations we were interested in were present in all of the samples. The program we use for that is called Swift FCS Combine. Once those samples have been concatenated, then we run Swift on the concatenate to produce a cluster template. So this is a normal Swift clustering run, and one of the output files, as we showed in tutorial 2, is a template file, and we can use that to assign any number of additional samples to the same clusters. And that's the third step in this procedure. We can take samples that may contain all 3 populations, only 2 of those subpopulations, and so on, and they can all be assigned to the same cluster template using Swift assign main. So the consensus samples are an important component of this procedure. Consensus samples are derived from concatenation of multiple samples, or if the number of samples that we'd like to select from is too large, we can take random subsets of data. We can downsample from a large number of initial FCS samples. It's very important to get this correct. If you leave out a particular target cell population, then of course that target cell population will not be represented in the cluster template, and therefore you won't be able to get any information on those cells. So it is critically important that all target subpopulations must be represented in the consensus sample. For example, if cytokine-producing T cells are the target of interest in your particular set of experiments, then the consensus sample must contain substantial numbers of these, enough to cluster. And for Swift, that means that we need as many cells as we have dimensions. So in a 20-dimensional experiment, then we need at least 20 cells per subpopulation for Swift to be able to find it. So a simple strategy that we often use is to combine a proportion of the events in all samples. This proportion needs to be chosen so that the file size doesn't get too large. Swift works very well with about 2 to 5 million input cells, and so we often aim for a concatenate somewhere in that region. For example, if we wanted to work with 50 samples to get a good averaging over the whole experiment, then we could take 50,000 cells randomly from each sample, concatenate those to give us a good size for sample to work with. More complex consensus samples can be designed for particular purposes. If you have some very specific questions in mind, then the design of the correct consensus sample may help you to focus precisely on that question. But this depends very much on the experiment you're performing and the question you're trying to ask. So we'll use a simple example to illustrate this procedure. The experiment that we will draw from is one in which we had two groups. They were human peripheral blood mononuclear cells stimulated either A with influenza peptides, or B, they were left unstimulated, they were just a DMSO control. And what we would like to know, our target population, is how many activated CD4 T cells produce interferon gamma in response to influenza. So the strategy would be to take a consensus sample constructed from an equal mixture of influenza-stimulated and unstimulated samples, so that we hope to get representation of both the stimulated and unstimulated subpopulations. So Swift FCS Combine is the program that we use for this. It's in the Swift folder. So as described in tutorial 1, open MATLAB, open the Swift folder in the left-hand window, and now type Swift _FCS _combine at the prompt in the command window. That will bring up the FCS Combine window, and this allows some options for concatenating the FCS files. First of all, you can choose between two methods of concatenation, either a certain percentage of events per file. This is currently set for 100%, but you can change that to any percentage that you would like. The alternative would be that you can choose a certain number of events per file. In this case, we could choose, for example, 666,000. Once you've decided on the number of events or the percentage of events per file to sample, then you move on to select the FCS files. You click the button at the bottom, and that guides you through the usual system interface, so that you can locate the files, select the ones that you wish to combine, and then start that process running. The output of that process is two new files. One of them is an FCS 3.0 file that carries the name of the folder that the files were in. It appends cct for concatenate.fcs. So that file is now a new FCS file containing the subsampled and concatenate files that you selected. The other file is a CSV file that contains the information about the file names and the number of cells, and also the index numbers of those samples within the concatenated FCS file, so that you can always go back and locate which events came from which file. Now, to prepare for cluster template assignment, once you have made the consensus sample, you then cluster that in Swift using Swift main as described in tutorial 1. Examine that file to make sure that any target populations that you're particularly interested in are well represented in that template. This template is very important because you're going to use this one template to analyze the rest of the experiment, so you want to be sure that it's a satisfactory template. If necessary, go back, change the cluster number, or change some of the analysis parameters and rerun it until you're satisfied. So the template file from this analysis, which is going to have the experimental name plus template.mat, can then be used to assign the events in any number of additional samples to the same set of clusters. To start this process, after generating the cluster templates, you place one or more Swift template files in the folder MATLAB Swift D template. So that's in the same subfolder as T. So the template goes in the the folder labeled template, and the samples go into the folder labeled A, and you can put any number of samples into that folder. We can put hundreds or even thousands of files in there, and that will still work fine. These files should be from the same experiment, or at least should have the same parameters as the files used to create the template. They can have different compensation matrices because compensation will be applied by Swift before assigning all the cells to clusters, but the dimensions must be the same for all of these files and they must match the template. So at this stage you can open MATLAB, double-click the Swift folder in the left-hand window to open it up, and then type swift underscore assign underscore main at the prompt in the main MATLAB command window. For samples with about 1 million events each, and assuming that we have a template of about 500 clusters and maybe 15 dimensions, then you might expect about 1 to 5 minutes processing time per file. So this process is much faster than the original Swift clustering because it simply represents the last stage of the normal clustering run, the assignment of cells to a template that has already been generated. The assignment results are placed inside folders within the A folder, and they're titled with the name of the template. So in this case we have the DMSO G2CCT template, and that name is put onto the folder that contains all the results. And within that folder there will be files, the usual Swift output files, all except the template file, and that'll be for every sample that is in the A folder. If you have put more than one template into the template folder, then all samples will be assigned to all templates, and the results will be output into multiple folders of this sort within the A folder. In a large experiment, Swift will produce a very large number of output files if, for example, you have hundreds or thousands of input files. So to bring that output data together into a form that's much easier to work with in a spreadsheet, we have a small program, Swift text combine, that can be invoked by typing swift underscore txt underscore combine at the prompt in the MATLAB command window. That will open up the program and open up a dialog box. So navigate to the folder that contains the output. So in this case it's within the A folder, and it's this folder labeled with the file name plus template. Highlight that folder, click open, and the program will then assemble all of the data from the text files in that folder into 4 combined output files. Those are summary tables of the data that are now very convenient to work with in spreadsheets. Looking at the information within those files, the merge cluster sizes or the split cluster size file, those are relatively simple. The first column lists the cluster number, and the other columns are each taken by one file, and so for each sample you have the number of events that have been assigned to that particular cluster. And you'll notice fractional numbers in many cases. This is because the assignment, as with the original program, is normally probability based, and so these are the probabilities rather than assigning just discrete cells to each cluster. There are 2 folders of that sort for the 2 cluster size sets of results, and there are similarly 2 more files for the merge medians and the split medians. These files have a similar format where the cluster number is accompanied by 1 column for each file and for each parameter for that particular sample. These clustering results can then be explored very easily using these tables. So you have a choice of using the Swift FCS files and exploring the output in a program like Flojo and performing gating and batching in those kinds of analysis programs, or you can use these summary tables listing the cell numbers and median values in each cluster, again using whatever analysis program you prefer to use at this point. So that concludes tutorial 3 on the use of Swift to prepare templates and compare samples. And I'd remind you that tutorial 1 and 2, if you haven't seen them already, tutorial 1 is installing Swift and performing basic clustering. Tutorial 2 is visualizing the output data from Swift. So thank you for working through these tutorials, and I hope you enjoy all the masses of information that Swift is going to give you.